Yeah, once again, thanks for the invitation, and I hope you will be happy with the update that I have prepared for today. I thought, because I think I'm the first non-clinician and non-geneticist really talking today, I'll talk a bit more about function and protein and tell you a bit how we study function, and then introduce you that model or the variant that we are actually studying using a mouse model, but also using patient iPS or induced pluripotent stem cell lines. To start just off, I think we already, I guess, learned most of this, but let me just introduce the protein. I think KCNQ2 as a gene, we already learned. That's the name that is sometimes also used for the ion channel, or often also used for the ion channel. It encodes, but that channel has also a different name that is maybe in some of my slides, just to make it more clear. It is called KV7.2. And you have heard from previous talks, and especially like broad range of disorders that have been now linked to KCNQ2 variants. And Sarah has mentioned in my bio that I did PhD in Germany. I actually did PhD in Germany on KCNQ2 variants, but at that time only linked to benign familial neonatal seizures. So when I wrote that PhD, I often had to emphasize that the reason I studied that, because it's benign and it is rare, is to have a model for other epilepsies. But it turned out, like everybody says 10 years ago, that KCNQ2 has a much bigger contribution to disorders, and we now see that that spectrum has really increased. I'll talk a bit about expression. So we know that obviously it should be, the ion channel should be found in brain if it causes epilepsy, and all the other co-expressions. I like that expression too. Then we also know that KCNQ2 normally doesn't come alone. It likes to hang around with a friend from the same gene family, KCNQ3. Either alone or together, they form what we call M channel, and that is because they give a brain type of current that is called M current, and I'll tell a bit about that too. So we've seen the schematic already, but just maybe to introduce, this is sort of important for us doing function, that we always talk about the ion channels or potassium channels having 4 subunits. So you see they need 4 of them to come together and make that little pore through which the ions will flow, and this is what we measure as the current. And when we talk about gain or loss of function, we mostly consider whether there is more or less current flowing through, so more or less ion flow through that pore. Now you see that the channel structure has all the 6 transmembrane regions, and we know that this S4, for instance, is the one that is sensing how the membrane voltage changes. So this is actually the one that receives the signal that the channel should open or close. Then we have the pore region that is formed by S5 and 6. These are kind of the ones coming together to make that little hole through which the ions will flow. And then this internal long C-terminus, I'll mention that again, seems to be a very specific characteristic of this type of potassium channels. And this is like their little antenna with where they receive all the signals from inside of the cell that tell the channel whether it should stay open, work more or less, and what else is there to be done. There is this orange guy here. This is just like a little helper, so we call it a beta subunit, and it also joins this complex of the 4 subunits. Now, there's been some discussion about how much has changed in the last 10 years, and I will just throw in here a few things that are now more available for us, and they have developed also for the functional analysis. For instance, there is this beautiful site that has recently become very interesting to many scientists, where you can find basically a predicted protein structure of any protein you can think of, and this is a KCNQ channel. So from that really simple schematic on the previous slide, you now see how the channel actually would work or look if you kind of take into consideration the folding and all the changes that proteins do, and you can see everything here in blue is sort of really predicted as is. And then there is a long part that is not. So all these like blue ribbons are actually the transmembrane regions or important functional regions, and we know a lot about those. And what you can do with the program is zoom in and find where your specific amino acid is that you're studying and see how their interactions or how it interacts with the surrounding, where it is, positioning between different ribbons and so on. So it's kind of one of the amazing tools that will surely help further advance our knowledge. Now, we want to know about the expression of channels, and we wanted to know that for a long time. And this is sort of one historical image I've never published from my previous life in Germany, where my colleagues did this in situ hybridization. So that was basically using brain slices. You kind of, one of the questions was, of course, how the channel changed the expression over time. So first where they are in brain, and then also how they change the expression over time. And if you look at these little images of sections of brain, you see wherever there is a dark spot or dot, that would indicate that you have some more that channel, and you can see that KCNQ2 and KCNQ3 are kind of spread through the cortex, and there is this hippocampal region that is also affected. So the little circular parts here, but then there were some differences that we saw between the KCNQ2 and KCNQ3. These are P3, 10, 20, and 40 are basically different days of life of a mouse where the brains were extracted. This did tell something, but in the end, like again, there are many advances in this field, and one of those is availability of single-cell RNA sequencing. We now have many studies that actually use tissues from either mouse or human tissue and sequence only what genes that are expressed in those cells. And we had a little small collaboration with bioinformaticians and took one of those databases from human brain and looked at if we can cluster those neurons or those cells that are identified in that brain tissue by their properties, by the gene expression pattern. And because we are mostly interested in brain excitability, one of the important things was to cluster them into excitatory and inhibitory neurons, but we also have other different cell types that are found in brain here, and then look at how different genes, and here I present KCNQ2, are expressed in those clusters. This just illustrates for you that we now know that KCNQ2 is actually more expressed in excitatory cells, but we do have inhibitory cell population that also expresses this gene. And just there's been recent publication in Nature that kind of combines sequencing data, electrophysiology data to provide a brain map. So I'm sure there will be even more advances from the time we have those maps and those atlases available. About the subcellular localization, you kind of want to know which cell types the channels are expressed in, but you also want to know where they are in that cell. And I already mentioned that KCNQ2 and KCNQ3 like to rather spend time together and interact. And where they actually found is at the axon initial segment. And axon initial segment within a neuron is a spot where action potentials are generated. And it looks like KCNQ3, which is shown here in the picture, so in red. That is the exponential segment part. That KCNQ3 is kind of like an anchor, so it brings KCNQ2 also in this position. But KCNQ2 can be also found within soma and dendrites. This is again one very old study, but again looking at the developmental expression, just to show that we do have KCNQ2 signal on the left very early in mouse brain. And then as the mouse gets older, if you look at the green here, you see that more and more of that green goes into this worm-like structure, which is actually again the axon initial segment. So the channel is expressed early, but seems to populate axon initial segment later together with KCNQ3. Now, I mentioned M current, and M current is brain current that was named because it can be blocked by muscarinic agonists. So basically there is a whole communication within a cell between two different systems. So muscarinic receptors can get activated, but that activation will suppress this current, and that is kind of like what happens regularly in brain, and that's how the current was named. And we know also before we knew that KCNQ channels are the ones responsible for that current, we knew that if the M current is suppressed, like in the picture on the right, if it's smaller, you will get more action potentials, more firing. So like reduction in M current increases excitability. So that was something that was known. And then how to study all these variants and all this genetic information that we get. And in case of single variants, the first thing you can do is, of course, put that gene and that variant in a cell. So you can either use, and even kind of, I guess, sometimes funny for people who don't work with that, frog eggs, and put the gene, wild type gene and mutant gene, and compare their properties. I'll show you some data. From there, you can just use some other cell lines that are used in regular and not really neuronal. So they're kind of the commonly used is the embryonic kidney cell. So you can also use neuronal expression models. That's kind of like special preparation of mouse neurons where you can put your genes of interest in and again study them, or make animal models that I will also talk a bit about. And then also a big change that has come in the recent or in the last decade is that more and more studies are now using induced pluripotent stem cells. So the way that you can use patient biopsies, skin or blood, and turn it into stem cells, and then turn those stem cells into neurons or other tissues that you want to study. So these are, I'll kind of talk about three different approaches here, and that all in relation to what we know is Australian variant, K556E, identified in Australian girl. I hope I summed up, I kind of just put a few lines here about the phenotype. This is considered the milder form of the, Ingrid mentioned that there are some milder forms, but we always kind of worry to use this milder term. And I'm sure Ingrid would have many more details, but we know that the seizures started early. They were quickly under control, but then their anxiety, autistic features, spontaneous rage attacks, aggression, and other co-expressions. And the variant that was identified is K556E as a cause of the disease. So K556E is found in the C-terminus that I already mentioned. It's like a little antenna and major communication center of KCNQ2 with the world, but basically mostly with what is happening inside the cell. And you see here this little A, B, C, D, they all have different functions and different molecules they interact with. And the variant is found basically at the end of this B part. There are some hypotheses that and things that could be tested. There are quite a few variants that are identified in the C-terminus. And that is why it is exciting to look at these different parts of C-terminus and what variants, when they're found in different regions, actually do. Also, often when you get that, like just from the functional point, you also want to know that your variant, not only where it's localized, is it conserved between different systems? So can you align mouse gene, or can you align other KCNQ genes and see that they all use the same amino acid at that position? And if that is the case, that also tells you that there was a evolutionary pressure to keep that cell because they play an important role. I'll start here showing you some of the data. So I mentioned already frog eggs. This is how they look, black and white pole, and this is like about 1 millimeter big cell. And you can record it with a method that we call two electrode voltage clamp. We have now systems that use plates, so you can put 96 of these eggs injected with different combinations of genes and then record the currents. And you see here examples of currents recorded from the wild type on the left, kind of seeing that it is like a very slow outward potassium current and doesn't show any kind of inactivation or closing. And here, you know, like see that it kind of just goes or gets bigger the more positive the membrane gets, which is sort of normal for potassium currents. And then when you compare that to the mutation variant K556E, you see that the variant is smaller. So the currents are smaller. You kind of can sum that up over different potentials. And what we sort of conclude from this slide is that the currents produced by the mutant were smaller, but not completely abolished, which is also commonly seen in KCNQ for KCNQ mutations. And this moderate loss of function didn't actually, there were no other changes, there are other parameters that you can analyze, and we haven't seen anything else in all sites, just this reduction in current amplitude. Now, we did use a different cellular model. We did use CHO cells or CHO cells, Chinese hamster ovary cells, just because they're kind of closer to mammalian, so they're mammalian cell line and closer to us than the frog egg. Strikingly, there were not many changes we could see for KCNQ to wild type versus mutant. You see that amplitude here. So if you just compare red with the blue, kind of red is a bit lower than the blue. So it could be a bit of a loss of function, but it's not really a big difference for this line. One difference that occurred in cells that was not so obviously or was not detected in all sites was this faster deactivation. So it looks like channels expressed, mutant channels expressed in CHO cells are actually closing faster. So channels open and close, and these just like shut the door faster. So that would also be a loss of function mechanism. We did look at parameters of the same expression, but when we combine wild-type KCNQ2 and wild-type KCNQ3, so with a friend, and there are not many or any differences that we detected between the two combinations, which was also interesting observation. Now we knew we had a partial loss of function and wanted to know what would this variant do to the brain, or what would it do within the whole organism, and that's why we generated mouse model. There are different ways to make those mice. So what we basically did is just to find the same position in a mouse gene and insert the same amino acid exchange on that position, and then looked at how these mice behave, what is their seizure phenotype, and look at also neuronal activities, so like look at what single neurons do. And then, of course, this might can be used to test drugs, especially if you identify any kind of difference or phenotype. Here are a few examples. So EEG recordings on the left, this is how they would look like when you record them from mice. I won't be showing any here because you will hear why it is a bit tricky to get those. From this mouse line, we did do, and I'll mention later some, but I will be showing more of analysis on a cellular level. So these are trains of action potentials discuss single neuron talk or work. So the first observation related to the mouse was that while they were, we started noticing that the animal carriers were leaving notes that often when they're changing cages, when the mice are being cleaned and fed, that they would occasionally observe a seizure, and after that seizure, death. Now, this graph shows both homozygous and heterozygous mice. Homozygous are the ones that carry variant on both of the alleles. You just get them through breeding, but actually heterozygous is the mouse that is corresponding more to human condition because it has one wild type and one mutant allele. And you see here that homozygous ones, when we were still getting them, they were living by about day 50, 60, but like, they would just get seizure and die. And that in a mouse world means that they're just severely affected, and that mice cannot tolerate this variant or presence of this variant so well. For the heterozygous mice, this was not so clear. So we would have often heterozygous mice that look completely happy and healthy. And then for some of them, occasionally we would notice those handling seizures. If you catch a mouse to do experiment, it would get seizure and often not recover from that. So you see here that many of these mice were like dying. This is, you know, the normal lifespan of a mouse is 2 years. So you see that it is sooner than that. I have here one seizure that was recorded in a homozygous mouse, and it will happen very quickly, but it is interesting because we know that we do not relate a KCNQ2 to SUDEP, but this has happened really fast. This mouse was looking like it is sleeping and then just like moved quickly, and this is already a short seizure and immediate death. So that was what was happening with the complete heterozygous, complete homozygous mouse, so mouse that has mutation on both alleles. Because this was such a severe phenotype, we actually were not managing to analyze more of it because it was difficult to breed them or to get more homozygous. And anyway, we said this is a more severe phenotype that we would expect, so that really correlates to the clinics. So we went to analyze more of the heterozygous mice. And this is a typical test we do when we analyze epileptic models, and that is the seizure susceptibility test. So you either use higher temperature to induce seizures, and it is known that if you do that, you would have, you know, even in a wild-type mouse, that increasing temperature that would cause seizure, and finally, you can measure the time that it takes to get those seizures, and you see that in red, the heterozygous mice are more susceptible, so more sensitive to the temperature. That doesn't mean necessarily that KCNQ heterozygous mouse would have febrile seizures. It is more that the brain is sensitive, so there is a disturbance or there is something happening in the brain that makes it more susceptible to increased temperature. The other on the right show the similar test that is done with a chemical PTZ, and you just like can induce the same effect, so the same increased susceptibility of the heterozygous mice. Now, another battery of behavioral tests was done because we know that all these things do come together. So in epileptic encephalopathy, you do have changes in behavior. And interestingly here, without going really into details, you see locomotor test, you see Alois plasmase test. These are done to kind of measure different aspects of mouse behavior. Light-dark test to see kind of the mice like to be more in the dark than in the light. But between the black and the red here, you don't see many differences. The only difference that we found in behavior was marble bearing test. It's kind of, if you give marbles to mice regularly, they'll try to hide them, so kind of bury them. And that was reduced for the heterozygous mouse. This test is not, it's really difficult to correlate what the behavior of a mouse really means in humans, but it could be an indicator of cognitive abilities. And that was, yeah, so I did say I will mention EEGs. We did some recordings, the EEGs, they were pretty clean, so very similar to the wild type. And we're repeating now those at different time points. Because we kind of think we may be missing a time frame when the electrical activity in brain has changed. But because of this handling seizures, so because when you kind of catch a mouse, it's sort of more sensitive and get seizures, it is not easy experiment, so it kind of still requires some time. Other approach that we used was then to get extract the brain slices, so section the brain and get then to the single neurons and then look at their activity and see whether the variant changes anything about the activity of these neurons. This was done in the cortical neurons, layer 2 and 3, and surprisingly didn't reveal any significant differences at this stage. So like if you just look at whether the potential of the membrane is, you know, the same. It looked like a mutation could be introducing a more positive potential, but then it was not significant. So we kind of were already looking into what is happening here. One good thing is that when we measure the M current, so carried by KCNQ2 and KCNQ3, together in these neurons, that we do see that it is reduced in size. So that's kind of indicator of that loss of function that I mentioned that the variant introduces. But that didn't translate into any change in the firing properties. So like if you look at these neurons, wild type or heterozygous, they all fire with the same frequency, and absolutely nothing changes in the properties of action potentials, which was really surprising to us. Now, I have to say, and I will mention that, these recordings were done a bit later in life, so it was 35 to 40 days of life of these mice. And we presume that is the reason that we don't see changes. We did go that late because that is where we started observing those seizures in heterozygous mice. I thought to give them more time to develop, and show us also what we see in terms of seizures, but that here didn't help. So to summarize this part, we saw that KCNQ2 556E variant that is causing a mild form of DE has a moderate loss of function, that the mouse model has spontaneous seizures, premature, that higher seizure susceptibility and some limited behavioral changes. We saw that neurons show smaller and current, but no changes in firing rates in cortical neurons at P40, some more work to be done there. However, from everything here, this mouse model does give us a few markers that could be used, disease markers that could be used for drug screen and, to test the efficacy of potential treatments. I will now tell you a bit more about iPS-derived models of K556E. So I already mentioned induced pluripotent stem cell, the technology that now is already 15 years old, but has started being broadly used in the last, I think, 10, 12 years. The labs that are using it are trying to get patient biopsies, materials. I think there would be many on this call that were or are involved in such studies. From the skin biopsy, you can use different methods to turn those cells into stem cells that can then become again anything they want. And of course, in terms of epilepsy, we want them to become neurons. Now, in terms of which kind of neurons they can become, we can grow them like just on a dish in like a flat culture, 2D culture, but we can also make what we call brain organoids, or like mini structures that sort of reproduces a bit better development of the brain. And then, of course, there are many things that you can do in terms of phenotypic analysis to study these neurons. So why are these important and why do we use them? It's because first we can get a patient-specific context. So I think also in terms of what Lynette was talking about, it is one variant, but there's some modifiers, how specific they are. And if you have a patient-specific line and just correct that mutation or make what we call isogenic control, that may be telling you something in at least in functional terms. You can have that information in terms of human biology context, because as much as we believe mice are similar to humans, or we are similar to mice, there would be many differences, especially in terms of brain development. And then, of course, that means you can really focus the analysis on early developmental processes, and then look at the study, look at the disease mechanisms, and of course, gene therapies. One suggestion is also to use these cells to do qualifying diagnostics. So if you have a treatment and not sure whether it will be effective for different patients, maybe looking first at the stem cell derived neurons would be a good way to go and test that. This is one of the protocols that we use, and this is probably very broadly used in recent years. It's called, let's say, MGN overexpression protocol, but basically you just put one transcription factor, so one molecule in the stem cells, and they turn into neurons within a week. And within 2 weeks, they start firing. So, well, this is sort of a forced expression or forced development of neurons. So it's really quick. I mean, neurons don't develop that quick. Normally, if you use the protocols that are following more the developmental pathway, it is nice model because it still gives you the neuronal context and you shorten that time that you need for the analysis. I will mention that time aspect, especially in terms of brain organoid analysis. One thing that we have done in one of our previous studies is to kind of estimate how does this protocol correlate with the brain development. And you see here sort of what happens in utero, in the womb, like in different processes that take place in terms of how our brain develops. And whereas, of course, there's some protocols out there that would cover most of these processes. The NGN overexpression, so the protocol we use covers a small aspect of it, but still provides some information about early neuron activity. So this is how the activity of these neurons look like. Again, you probably recognize now action potentials. In blue here is the wild type, in red again K556E patient line, and wild type line actually here is a control, population control that is age and sex matched. And we see immediately that the cells obtained from patient line are actually firing much more. So you see here increased frequency of firing, which is sort of very convincing result. You can analyze many aspects of this. One thing I should mention is that we did these recordings in these neurons because I said they can start firing from 10 days to 2 weeks after you started making them. But then they do have a bit of developmental pathway and kind of stabilize the activity after 3 weeks. So we looked a bit earlier just to make sure that we don't miss that early onset that is often mentioned in relation to KCNQ2. So that's why this is like after 2 weeks, and we already see this increased excitability phenotype. And then the same was repeated with, sorry, I think this is, yeah. And the same was repeated after 3 weeks, and you see again that we have increased excitability. So that kind of confirms that the NGN neurons nicely reproduce at least what we expected that KCNQ2 variant would do. So reduced M current would give increased excitability, and we see that in these NGN cultures. However, what we don't see, and one other thing that I would mention at 3 weeks is that we did see some significant differences in capacitance, which actually could suggest, so capacitance kind of a measure that we obtain from patch clamp recordings, but it depends also a bit on the size of the cell. And we think in NGN neurons maybe also on how fast they're developing. So like it can be a small indicator that actually the variant and what is happening in the cells at this point could be translating also in some subtle morphological changes, which would be interesting to follow. And to look into those, I mentioned organoids. I don't have any data for this variant yet from organoids, but I did want to mention them and show you that there are different ways to make this little ball that can stay then in culture for a long time. Many studies that have been done, and there are many, let's say, cooking protocols at this point, have shown that the best way to analyze them is from 3 months on. So you see that the advantage of NGM protocol is that you can get those results faster in terms of screening. But in terms of developmental processes, this is probably the way to go. We have also, so this is just like if you section those organoids, you see that they have some structure inside, and you can then look for your molecules and also look for KCNQ2 and which cells and where it is found. And the other good thing is that these little balls, and you see now how one looks on the left, can also be used for functional analysis. So you can load these cells with dyes and then measure calcium activity, which is actually the indicator of neuronal activity. But what this gives you is that you can also look at how these neurons talk to each other and how they connect to each other. And then in terms of morphology, of course, how is this brain organoid organized between, like when you compare wild type and heterozygous condition. So I think I've shown that patient-derived NGN neurons did reproduce loss of function of K556 and that increased neuronal firing can be linked probably to occurrence of seizures. We don't know how it links to other features found in patients, that there may be some potential difference in cell maturation morphology, and that, of course, having these NGN neurons that have a difference between patient and mutant line gives a chance to, again, use it for screening therapies and looking into potential drugs or medications that can help this dampen this difference. And then, of course, that further investigation, when we would use also more of expression analysis and morphology analysis and network analysis, will probably be very useful. I kind of think we will be getting there, you know, with one by one mutation. So I know that this study was focused a lot on one variant, but I think this is sort of how it's done with many of them. And this is the interest in this one wouldn't, you know, just like this is what we had to focus on, so kind of have to start somewhere. I'll finish with this slide from the student who did most of these studies, and the quote from Steve Jobs. I think that there is a genetic information missing here between the patient and the oocyte, but sort of shows that you know, we learn a lot as we go, and that all these functional aspects that we do can help develop therapies that are sort of targeting really what the cause of disease is. I want to thank, in the end, people involved in the study. So we have Yafit and Lucas, who did most of recordings, and Cherry and Nicola, who supported them, of course, Steve and Ingrid, and everybody from the team, and patients and parents. And thank you very much.
Thank you so much, Professor Maljovic. A couple of questions that are coming up in the chat and elsewhere. You know, you mentioned there at the end that this was a focus on one individual. If the models are made with the specific variant of the KCNQ2 mutation, I guess the question is, how broadly relevant are they for therapies for others as well? Do we need more models, and do they need to be mouse models, or do they need to be different kinds of models? What do you think?
You know, I showed that scheme at the beginning, and I think there will be probably talks tomorrow. We do try to understand the mutations or these variants and functionality, not in terms of single amino acid exchanges, but how they actually affect the whole domain or how they affect the whole molecule. So I kind of think there would be shared things between different variants that are found in specific region. And I think that that's where we, you know, we can't make model for every single variant at this point. Maybe at one point in the future, we will be that fast and have everything modeled. But I think it can be really relevant for instance, all the variants that cause similar effects. So that's why, you know, we kind of try to select them from different regions. We know already a lot about core mutations. There are models showing that they have dominant negative effect, or that S4 mutations can have gain of function, or that, so we kind of have them, or like shift in voltage dependence in both direction. So we have a bit of idea how different domains of the molecule work. And I think each of these models also help us understand the biology of the channel protein more, which of course in turn becomes relevant in terms of any mutation, because, you know, we have to kind of have to understand. I did try to say that we still don't know so much about these proteins. We kind of, you know, talk about KCNQ2 a lot. We know how it's structured, but we don't exactly know everything about its expression pattern, its molecular interactions. I mean, there is more and more every day, but like each of these variants studied can actually help understand that better, because that will, you know, indicate some other biological functions in the cell that can be important for the function of channels.
Let me carry on if I can, Snez.
Yeah.
How do you compare the M current? You talked about the M current. How do you compare the M current results in mice versus humans, given the genetic differences and the importance of genetic background? Is that a challenge?
Well, we kind of think that they have the same function. You know, sort of the M current was described first in some kind of cellular model, and you know, from mouse or rat, and then you kind of, you know, a lot to describe there, and then sort of you try to find it or identify that it has a similar function in brain. So it is not. So there are differences in genetic background, but the channel seems to play the similar role. You know, the brain how to say, excitability paradigm is similar in mouse and human brain. Of course, there can be differences that are related to specific interactions or very tiny functional impairments, or that will look different in a mouse brain. And we think, for instance, this mouse model doesn't show much of phenotype, probably because mice tend to compensate a lot. They kind of change their other genes to just like feel, and keep the brain and the system working. And you often see that when you introduce this very severe variant. So we know that there is no absolute translation, but it still is, how to say, in pharmacological terms, in terms of what we understand it currently, it looks pretty similar between the two systems. It's sort of like, this is M current is like what kind of we have, like we know that it works as a current that can suppress repetitive firing and help keep the membrane potential at a certain level. This is sort of kind of universal across at least higher species.
Speaking in a kind of a slightly broader sense now, this is really interesting science. How do you envision it moving forward to help patients in terms of developing treatments, ASO, small molecule treatments and the like, gene therapy? How do you envision this and other mouse models helping in terms of patient therapies and providing treatments for kids?
I'd say, obviously, I guess from the title and everywhere, I kind of try to put IPS lines more in there, because I do think that human background, it does matter, or patient-specific background does matter. And I think, especially like as far as I know, in KCNQ2 field, there are many repositories of IPS lines or fibroblasts that can be used to kind of look at those mechanisms. We do not know much about that yet. We do have several examples or several studies that have been published, but while human lines are then useful to sort of understand a bit more of human biology and maybe like what is specific for patients, mouse models are still have advantage that we do have to look at the whole system. We can look at the whole brain. We can't do that with mini brain. It's kind of just a mini brain, and that effect on the system is important to see through the mouse. So as I said, like the more mouse models we have, probably the better. But I still think that if we manage to get them from the different functional parts of the protein, that would already cover or indicate, that we are, that they all share the same pathomechanism, even though the mutations may be very private and specific. And in terms of development of drugs, I mean, we still know, like you can do the screens in human cells or cell lines, but important preclinical models are still mouse models. So providing any kind of proof of efficacy in a mouse model is still critical.
Okay, one other question in terms of that, you mentioned in your study, you talked about a couple of aspects in which function was impaired. You talked about, for example, the hiding of the marble. Are those ways in which through mouse models, we might be able to, whether this mouse or another, move beyond the notion of simply looking at whether or not it's going to affect seizure control and look more broadly at behavior and by extension development?
Yeah, I think that was the idea, adding all these behavioral sets of experiments. There are even more than that. Then also like looking into morphology, I think it is, becoming more and more obvious that even when subtle, like if we are able to look at the expression patterns and changes in expression patterns because the mutation has affected the brain in a certain way, and then the brain is responding first by seizures or less by seizures. I mean, it's really interesting with this model that we basically have two populations of mice in that heterozygous group. Some heterozygous look happy and don't get seizures at all, and some get seizures and die earlier. And they have the same variant and they're crossed in the same background. So the kind of thing is happening is that there is that instability in the brain that just for certain reason expresses more in one group of animals than the other. It's really confusing that it happens in, animals with a very preserved background. But just looking beyond seizures, looking at behavior, looking at expression profiles, looking at morphology will of course be important to give insight into complex disorder like epileptic encephalopathy, because it does have many other co-expressions or other things happening, not only seizures.